Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and we're going to continue our lesson today talking about the central dogma of biology. Today we're talking about something very important. We're talking about DNA replication. So every time we make new cells in your body, each of those cells needs its own copy of DNA. And this is the process of DNA replication. This is how we can make extra copies of DNA so that every cell has exactly what it's needed. There are a couple of complicated steps here and some enzymes with some funny names, but just hang in there, follow along to the best of your abilities, and I'll make sure that you understand this the best that you can. So thank you so much for making it, and we'll go ahead and hop into the lesson today. So let's talk about DNA replication. As a reminder, DNA replication is just how we make extra DNA molecules. Every time we make new cells, those cells need extra DNA. So these are some of the steps that we'll follow when we're doing this. When your body needs to make new cells, one critical step is to replicate new DNA. Each cell needs its own copy of DNA, okay? So it needs that DNA in order to function properly, okay? Copying DNA is a complicated process, okay? The DNA must be separated down the middle without damaging it, so that's the first thing that's gonna happen. Then each individual nucleotide must be copied. The newly created DNA strand must be proofread, then the DNA strands must be sealed back together. Okay, so we're going to talk about this process in a lot of detail. What it's saying right now is that when you have that DNA molecule, the first thing that you have to do is unzip it and make those nucleotides exposed. Then we're going to start adding in new nucleotides, and then when we're done, we'll seal it back together. Okay, so that's kind of some of the steps involved in DNA replication. Okay, in a DNA molecule, the two strands are said to be anti-parallel. So look at those arrows on the side of the board. You see they're parallel, so that means they're side by side, but they never touch. But they're anti-parallel, which means they're running in opposite directions. So you can see that one of the arrows is pointing up and one of the arrows is pointing down. We would call that anti-parallel. They never touch, they are right next to each other without crossing, but they are facing in opposite directions. Almost like how you can drive like a road where the traffic can go on both sides of the road. Okay, so the strands are oriented in opposite directions. And because of this, one of the strands is going to be a lot easier to replicate. The easier to replicate strand is going to be known as the leading strand. What happens here is we can just replicate the whole DNA molecule at once. The other one, the strand that's harder to replicate, is known as the lagging strand. And I'll show you why it's more complicated here in just a little bit. Basically what's going on is that with the lagging strand, instead of one long molecule, it has to be made in a lot of short strands. Those short strands are known as Okazaki fragments, and then they're linked together to make a full strand. Okay, so I know it's a little hard to visualize right now, but we'll look at a picture and label some pictures here in just a second, and it'll make more sense then. Okay, so more information about DNA replication. Okay, DNA replication is said to be semi-conservative. Okay, what that means is that when DNA is created, one of the strands is new and the other side of the DNA molecule is old. So we've conserved some of the DNA. Here are some of the important enzymes that you need to know, okay? I'm going to give you the name of the enzyme. I'm going to give you a little mnemonic way to remember what it does, and then I'll give you the full job in just a second. Okay, so one really important enzyme you need to know about is called helicase. Okay, the job of helicase is that it unzips the DNA molecule. The word I want you to remember is hacks, because to hack something means to split it. And hacks and helicase both start with the letter H. So helicase, its job is that it unzips the DNA molecule. Just think of the word hacks, it hacks it in half. Okay? Another really important enzyme here is called primase, okay? DNA primase, it primes these other enzymes. To prime something means to get it started. And that's kind of what this enzyme does, is it kind of lays down like a starting line so that the other enzymes know where to start, okay? Another really important enzyme is called polymerase. You can have DNA polymerase in DNA or RNA polymerase in RNA. its job is that it's adding all these new nucleotides. When we're copying DNA, we have to assemble the DNA nucleotide by nucleotide. The polymerase is the one that's adding those nucleotides. The word I want you to remember is that it pastes. Almost like when you copy and paste something, you're putting letters there. That's what polymerase does, is it's adding all these nucleotides. And then the last one that you need to know is called ligase. Ligase, its job is that it takes all those Okazaki fragments that we made on the lagging strand and glues them together. And so the word I wanted you to remember for this one is links. It links them together and kind of ties, make sure all those short fragments known as Okazaki fragments are linked together to make one long DNA molecule. So here's some more information, and then we'll get into that picture in just a second, and that'll be a little bit easier to kind of orient yourself. Okay, so here's what happens when we're replicating DNA. Okay, so replication of DNA happens in 3 steps, and you're going to see these 3 steps a lot. The first step of DNA is called initiation. DNA replication is an all or nothing process. What that means is that when we start to copy DNA, it cannot be stopped until the whole thing is done. It's all or nothing. Okay? The next step for DNA replication is known as elongation. Elongation basically just means to make something longer. Okay? If you were to stretch your arm out as far as you could, you would be elongating your arm. So in this process, DNA polymerase pastes complementary base pairs to the exposed nucleotides. So it's basically making the new DNA strand. Okay? And then the last step of DNA replication is known as termination. To terminate something means to end it. So the last step of DNA replication is termination, or just to end it. What's going on here is that once the new strand of DNA has been made, the RNA primers are replaced with DNA bases, and ligase links nucleotides together. Okay, so here are some more things that are going on in DNA replication. The first thing that happens is that helicase has to hack or unzip the DNA molecule by breaking the hydrogen bonds between the nitrogen bases. Remember, those hydrogen bonds are really, really weak bonds. They're strong enough to hold the molecule together, but they're weak enough to be broken by this enzyme, and that's a good thing. Then, once the DNA molecule is unzipped, we have a bunch of nucleotides that are exposed. DNA primase is going to go and lay down a primer. It's actually RNA. It's going to lay down an RNA primer, and that's our starting point, okay? And then DNA polymerase, it finds that RNA primer, and that's its starting point. It's going to start adding complementary base pairs to the growing chain, okay? Then DNA polymerase cannot start the sequence. That's why it needs the primer. So DNA polymerase doesn't really know how to start. It only knows how to, it can make a lot of DNA molecule, but it needs a starting point. And that's what the DNA polymerase does. The DNA primase lays down a primer, and then the DNA polymerase starts at that primer and starts to copy the DNA. Then DNA polymerase, something weird about it is it can only read and add nucleotides in one direction, and that's why we have this leading and lagging strand that I was talking about earlier. Okay, so now that we have gone through a lot of those steps, let's look at this picture, and hopefully that'll kind of orient yourself a little bit, and it'll make it a little bit easier on what's going on. So I'm going to kind of walk through this. I'm going to be pointing out some of these important enzymes in the order that they're happening in. So just make sure you're following along, and that way it'll make it a little bit easier as far as what's happening here. Okay, so the first thing that happens is this enzyme right here, this blue triangle. This is helicase, okay? Its job is that it's unzipping this DNA molecule. See, this is what the DNA molecule looks like most of the time. But when we're getting ready to replicate it, it has to be unzipped. So the first thing that happens is that helicase, it hacks the DNA molecule in half. So it's moving down this way, it's breaking these hydrogen bonds, and that's how we're getting one strand up here and one strand down here, okay? Some other enzymes are playing an important role here. You don't really need to know these. The purple enzymes right here are called single-stranding binding proteins, and then this green circle is known as topoisomerase. Their job is just to keep the DNA molecule, once it's unzipped, we don't want it to snap back together until we're done with it. So these keep the two strands of DNA separated for us, okay? So helicase starts to move through the middle of the molecule. It's unzipping it. It's breaking those hydrogen bonds. So we have one strand of the DNA up here and one strand of the DNA down here. The first thing what's going to happen next is we get this enzyme known as DNA primase, and it's going to lay down a primer or a starting point. You can think of it as a starting point. So it'll be a little, maybe just 2 or 3 nucleotides, and this primer tells DNA polymerase, hey, let's start here. Let's start the replication process here. So DNA primase lays down an RNA primer. Then the DNA polymerase will start there and start to replicate it by adding complementary base pairs. What I mean by complementary base pairs is, remember that adenine and thymine go together, and cytosine and guanine go together. They're complementary. They go together, okay? So we're going to start copying those DNA nucleotides. We're going to add the correct complementary base pair, okay? Then, this is the lagging strand up here, and I'll tell you how I know that in just a second. Once the DNA polymerase has added these nucleotides, we have a lot of these short fragments of DNA known as Okazaki fragments. This next enzyme, DNA ligase, is going to link them together. So it's going to move down this way, where these two strands are touching, it's going to link them together, okay? So what you need to know, helicase hacks, it splits the molecule in half, okay? DNA primase lays down an RNA primer, that's our starting point. DNA polymerase starts at the primer and starts adding complementary base pairs, and then DNA ligase links it all together, okay? A couple things I need you to know. We have a lagging strand here and a leading strand here. What's going on is that this enzyme, DNA polymerase, can only add nucleotides in one direction. It can't go both directions. So what's happening is when we're moving where DNA helicase is, where it's unzipping the molecule, we call this the replication fork. It's kind of like a fork in a road where you can take either side, but you have to take one. That's called a fork in a road. This is called a replication fork because that's where the road kind of splits. So one of the strands, if we're moving towards the replication fork, we can make this in one long continuous strand. And that means that we only need one primer, we lay down all of them together, and then we link them together and we're done, okay? The leading strand is a lot easier to copy. If you're moving away from the replication strand, so this enzyme up here has to go this way, if you're moving away from the replication fork, you have to make them in short segments. So you'll make a little bit here, and then more gets unzipped, and then you come back and you do it again, and then as it unzips more, you do that process again. So basically what's going on, the lagging strand, it's made discontinuously or in a lot of small fragments. Those fragments are known as Okazaki fragments, and those have to be linked together at the end. The leading strand is the easy one. You can make it continuously, which means you just make one long strand, and it's a lot easier than the lagging strand. Okay, great. So, couple more things. Here's that information that I was just talking about. Because DNA polymerase can only add nucleotides in one direction, we get a leading and a lagging strand. The leading strand replication is said to be continuous. It's one long strand. For the lagging strand, we say that the replication there is discontinuous or non-continuous because it's made in a lot of short fragments, and then all of those fragments have to be linked together at the end. Let's talk about that replication fork. Again, where helicase is separating the two DNA strands, that's known as the replication fork, okay? And eukaryotes, remember eukaryotes are, you're a eukaryote, organisms with a nucleus are eukaryotes. Our DNA is really, really big, it's really long, and so because of this, to save time, we'll get a lot of replication forks happening at once. We don't just do it all the way through. We have a lot of these replication forks happening all at once, and this is going to make the replication a lot faster. Okay, then here's the last step. So once we've replicated it and we're ready, we'll move into the final step of DNA replication, which is known as termination. Okay, what's happening here is we have to proofread the DNA molecules that we made. DNA polymerase is extremely accurate and makes a mistake. It does not make very many mistakes at all. Okay, 1 in 10 million. Okay, so that's very efficient. Okay, some polymerase have proofreading abilities, so once it lays down the nucleotides, we'll start to proofread it. Okay, once the nucleotides have been laid down, ligase can come and link the bases together, and at the end we have 2 new DNA molecules. One strand of each molecule is new, and one of each strand is old, and that is said to be semi-conservative. So if you look at the pictures of the arrows on the right over there, look what we started with. We started with 1 DNA molecule. They're anti-parallel, so the arrows are pointed in opposite directions, and the older strands here were designated as being rad. Okay, well, through DNA replication, we make these new DNA strands, but they're linked up with an older strand of DNA. So this is what we start with, where both of the strands are old, and then at the bottom down here, you can see that the 2 new DNA strands we have made, they're semi-conservative. One part of the strand is old and one part is new. So that's what the term semi-conservative means. Okay, y'all, great work on that. I know this stuff is really confusing. We're going to go through and label a diagram together real quick, and I think that's going to kind of help us figure out what's going on here. So teacher, go ahead, pass those worksheets out for everybody, and then when y'all are ready, come back and unpause the video, and we're going to work through it together, okay? All right, everybody, hopefully that didn't give y'all any problem, but just in case it did, I'm gonna walk through this with y'all. So feel free to add or change anything you don't have, and by the end of this, I hope that clears up some confusion for you. So number 1 says, how does genetic information flow? Again, this is referring to something known as the central dogma of DNA. It's the unifying theory of all biology, okay? Genetic information flows in one way. It goes from DNA to RNA, to proteins, all right? And we're gonna talk about this process in a lot of detail as the lesson goes on. All right, number 2 says, match the following nitrogen bases based on Chargaff's rule of complementary base pairs. Okay, so adenine pairs with one of these other 3 nitrogen bases. What does it pair with? Adenine always pairs with thymine. I just remember the word at, because those 2 letters go together, all right? Cytosine usually pairs with one of these nitrogen bases. What does it pair with? Cytosine pairs with guanine, okay? Now that we have those 2 done, these other 2 are gonna be a lot easier. Guanine pairs with cytosine, okay? And thymine pairs with adenine. Great. All right, let's keep this going. Number 3 says, match the function with the enzymes. So again, think of those mnemonic devices that I gave you. Number 1 says, unzips the DNA molecule. What hacks the DNA molecule in half? Helicase is the one that unzips it. All right, what links the nitrogen bases together? So the word link and ligase both start with the letter L. So that's how I remember them, ligase links. All right, which of these 3 adds new nucleotides to the growing mRNA? This one's going to go with the polymerase, okay? And then what makes a starting point for DNA polymerase? That's going to be our primase up here, okay? Great job. All right, let's keep this going. So number 4, put the following in order by writing the numbers 1 through 4 on the blanks provided. So this one says DNA polymerase adds complementary bases to the growing chain. Okay, so we're putting all 4 of these in order. So let's see what happens first. And this picture might kind of help us. So before we do anything here, look at this picture. We got to unzip the DNA molecule first, right? So anything up here about unzipping? Okay, so DNA helicase unzips the DNA molecule. So that's our first step there. Okay, great. All right, from there, the next thing that happens is the primase is going to add a starting point. So that's going to be number 2. All right, once we have our starting point, DNA polymerase can just add nucleotides to the growing chain. And then lastly, we need to link those nitrogen bases together using ligase. So that'll be number 4, like that. All right, here's what we're doing next. Here's that picture that we were talking about. We need to be able to label this and figure out what some of those important enzymes are. So let's see if we can figure out what's going on. Let's kind of do them in the order up here. So this says DNA helicase unzips the DNA molecule. Do we see anything up here that looks like it's unzipping the molecule? To me, number 2 looks like it's unzipping. So this is going to be helicase. Okay? Helicase is unzipping that DNA molecule. This is called the replication fork. Okay? Great. All right, number 2 here says DNA primase makes a starting point for DNA polymerase. Okay, so I see our primer here. What just laid that down? Well, that would be DNA primase, which is number 1. So DNA primase. That's laying down our primer. Okay, once the primer has been added, we're going to start putting in the complementary base pairs. So number 4 over here is going to be the polymerase. And then lastly, the ligase is going to come behind and link all of those nucleotides together. Y'all, thank you so much for following along. I hope that cleared up some of our confusion. We're going to talk about this in a lot more detail. Your teacher has some work for you to practice on, so make sure to give that your best effort, and I hope everybody has a great day.